Yes. Okay, here we go.
Carlos, I have a question. Who can see the questions in YouTube?
If you prefer, I follow the question used in the YouTube, and at the end of the seminar, if there is one question, I ask.
Okay. So
do you have my screen now?
Yes. Yeah.
Okay, perfect.
Yeah. So if you want, we can start. It really is my pleasure to introduce Dr. Shima Safaiyan. She is from Iran. She got her bachelor and master's degrees in microbiology. Then she moved to the Max Planck Institute for Experimental Medicine, and under the supervision of Professor Michael Simmons, got her PhD in neuroscience. Then she moved to Munich to do another postdoc. After her first postdoc working on microglia and cellular response to myelin and normal aging. And now she's working on microglial cell dynamics at the Max Planck Institute of Immunology and Epigenetics, where she's right now affiliated, really working, trying to understand in more detail the role of different subpopulation of microglia in the brain, and especially in the aging brain, which is a very interesting topic for everybody, because as you know, we are getting older and we need to know how our brain works and how to maintain it healthy, to have a healthy aging. So once again, Shima, thank you very much for accepting the invitation and for sharing your very interesting data with us. Please go ahead.
Thank you. I also say hi to everybody, and I think it's there the morning. Good morning, everybody. And yeah, I'm also happy that I can share some of my data with you in Mexico. I've never been in Mexico, but I would love to go one day. Yes, as Gustavo nicely introduced, now I'm located in Germany, and I have been here since 2011, and the story of my scientific life here has been always in aging, normal aging, basically. I didn't really work in the disease or pathological condition much. And the title that I chose is quite a general title for today. It's Myelin Microglial Contact in Brain Aging. This work is basically two parts. But the first part it was done during my PhD, and it was in the Max Planck Institute, and we could publish the data in Nature Neuroscience in 2016. And the second part, which today I focus mainly on the second part, is basically my first postdoc work, which was done in German Center for Neurodegenerative Diseases in Germany, under the supervision of Professor Michael Simmons. His lab is basically a myelin lab. He's quite an expert in myelin. And and the second part was also published recently in 2021 in Neuron. I'm happy today to introduce you what we discovered in aging with myelin and microglia. First, I want to start with the two keywords, as you can see in the title, myelin and microglia. First, it's good to know what is basically myelin and what is microglia. I will start with microglia. Basically, microglia are the resident immune cells of the central nervous system. They are very well known to be a guardian of the brain or central nervous system because of the specific shape or cell body that they have. They have a very elongated and branched processes, as you can see, and these processes constantly moving, even in the steady state in the brain. And with this movement, they are always basically, as you see, here it's showed, they are basically surveying the environment and basically touching, making contact with different structure, with different cells, and see if everything is okay. And then with the different activities and functionalities that they have, they basically take care of the whole environment of the brain. This is why they are called like surveillance microglia, or guardians of the brain. And another interesting things about microglia is that they have a very common gene profile with macrophages as the peripheral immune cells. Yeah. But they are also quite different. For example, they have the ability to self-renew themselves. The cells in the brain, microglia cells, they are not substituted by the bone marrow driven or derived macrophages, for example. These microglia are proliferating slowly, and they basically, with time, they also become aged, and this is very interesting. They are like neurons, basically. Although macrophages, you know that they are not like that. They always proliferate, they react, they die, and again, they get renewed quite fast. Yes, this is another characteristics of microglia. And yeah, microglia has been discovered that has different functionalities in the brain. For example, one of them is that they are in contact with astrocyte, and they are important for the maintenance of the integrity of the blood vessels. I don't go through that, just for you to know that how important are these cells in the brain. The other very important function that microglia are shown to have is that they are important during development for pruning the synapses. You know that synapses are very important for our cognition. In the presynaptic and postsynaptic neurons are quite, this connection is quite important for the impulses to move around and to make us cognitive, whole organisms. And the interesting thing is that microglia recently were shown to be important in this process, because they can, with the processes, we call these branches normally processes of microglia. In English can be a bit confusing. But anyway, these branches are always, can be in the development, they are in contact with the synapses, and they check, basically which synapses are weak or not good enough, therefore they can take them, eat them, basically remove them. Or also they somehow important for the neuromodulatory function around the postsynaptic and the presynaptic neurons. And one shouldn't forget that microglia are phagocytic cells. They like to eat, actually. And what they do with this function, they go around, and when there is some cells, especially neurons normally, they are dying and they become apoptotic. These apoptotic cells can be taken up or phagocytosed by microglia. Yes, these are the all functions that has been discovered within the time. And this is just a review that I put here. But in 2016, we could also show that microglia, through their phagocytic activity, are also involved in the clearance of myelin fragments in the normal aging. I mean, we showed that in the normal aging of a mouse, the microglia can take up the myelin debris, which are already accumulating in the aging tissue. And here I am going to introduce you the second keyword of my talk, which is myelin. What is exactly myelin, and why it's important to study myelin? Of course, myelin is very important for our cognition. And here I can tell you that oligodendrocytes in the brain are the cells that can produce myelin sheath. They are called sheaths around the axon, and they are basically, myelin is basically a multilamellar membrane that are stacked on top of each other, and they get they wrap basically the whole axon of the neuron. And if you take a look at the electron microscopy image from the cross section, you are now looking at an axon from above. Basically, you see an axon in the middle, and then you are seeing these black lines on top of each other, and these are basically myelin membranes. And this compaction of myelin membrane is very important, and there are two important molecules that are making this compaction, especially myelin basic protein, which we always call it MBP, and also PLP. Therefore, as you can see, myelin is some lipidic, consists of a lot of lipidic membranes on top of each other, and between these lipidic membranes, this MBP basically is pressing them together, and the compaction happens. And this compaction is quite important because through that, basically it's insulating the whole axon and leads to the normal conduction of the nerve fibers, meaning that a nerve impulse only can move fast and organizedly, only can move when the myelin is compacted. The internodes, which are here, basically are compacted, and then one signal can jump through the other node, and this basically should happen very fast to reach to another neuron, and the nerve impulse can happen. Yes, but as you see, the myelin is quite important for our cognition. If we don't have it, we cannot really, the neurons cannot communicate, and of course the body will deteriorate. And this is what is happening in aging. In aging, we slowly start to lose our cognition, yeah? And this means that in aging, myelin is quite affected. There have been some studies in monkeys, and there they already showed, again in the electron microscopy, just for you again, each of these roundish structures, which are surrounded by the black structure, they are axon, and this black structure normally in EM comes out like myelin. They are basically myelin sheets again on top of each other. Therefore, again, this is a cross section. You are seeing everything from above, yeah? And what you are seeing in an aged brain of a monkey, you see that, for example, some of the big part of the myelin is absolutely tears, basically tears apart. And then you have a lot of inclusions in this part, and this part is not healthy. Or you have some balloon in this axon again, or in the myelin sheet. And sometimes also you see that the myelin sheet is basically growing out. It's not really normal again. Or the compaction is gone. As you see, the layers of the layers that I told you, they are not really compact. They are now a bit tilted or somehow, yeah, further from each other. Yes, therefore these changes in aging is quite drastic, and this cause or leads to generation of a lot of myelin debris or myelin fragmentation in the brain. This is why in the mouse brain, we looked at the corpus callosum, where there are a lot of myelin around the axons. And there we could see in the normal aging that interestingly, again, in the axon, in the whole axon sheets, normally the whole axon sheet comes off and tears outside, and then sometimes this is basically completely outside and get released into the extracellular space. This we call fragmentation of myelin. That with time, it seems that myelin get fragmented, basically myelin sheets. And sometimes we could also see that these fragments and these myelin fragments are internalized into some other cells. Our quantification showed that the number of these extracellular myelin fragments is increasing with time, going up. That was very interesting. Therefore, we wanted to know, basically our main question or main goal was to know how does the clearance of this unstable membrane that I just showed you in the aging brain is happening in the central nervous system. What is happened when the membrane is out? It should be go away, because this is somehow a toxic structure, toxic substances in the brain, and should be released, should be removed. What happens to this debris? With that, we hypothesize that if we want to look at the removal of unstable membrane from the myelin sheets, we thought that according to EM studies that we had, and I showed you before, is that one big part of myelin sheets sheds into the extracellular space and makes a myelin fragment as it is here. Okay, now we have a myelin fragment. This myelin fragment cannot stay there, actually. It has to be removed. And of course, one of the best candidates that one can think and hypothesize that is involved in this removal functionality is microglia. As I said in the very beginning, they are the main phagocytic immune cells in the brain. Yes, therefore we hypothesize that microglia are the ones that are taking up the myelin fragments. And interestingly, when we did immunohistochemistry, and we could label the cells in red with a molecule which is called IBo1, and we could also label the myelin with myelin basic protein, as I showed you, the one that is important for the compaction of myelin layers, and we call it MBP. In almost whole my presentation, you will see a lot the immunohistostaining with MBP and also IBo1. Therefore, you always are visualizing microglia in IBo1 positive cells and myelin. What we discovered here, we saw that microglia with their processes or with their branches are often in contact with these myelin particles, which are basically a bit broken. And interestingly, we could see that this contact of myelin processes with, microglia processes with myelin is also again increasing with time. This means that microglia have a role in, they might have a role in removing the myelin debris. We took a closer look, and we saw that not only microglia are in contact with myelin, but also they can take it in. Basically, they can phagocytose the myelin debris. As you see, there is a piece of myelin labeled with MBP inside the cell. And again, this we could see only in aged animal, like from 18 months of age. And this basically the number of the cells which are having internalized MBP is increasing with time. Then here we could see that with time, with aging, that the myelin fragments number is increasing. Also, the myelin microglia contact is also increasing. I am not going through the details, otherwise it will be a very long talk. I don't want to make you tired with it. Therefore, I just make a summary what we have done and what we concluded at the end. First of all, we showed, or I already showed you kind of with some data that microglia are important for the clearance of myelin fragments that are accumulating in the brain. Yeah, they take it in. But the second part of this project was very interesting, is that meanwhile that microglia are involved in the removal of myelin, basically this the lysosomal system of microglia become overloaded with myelin, and this overloading of lysosomal system leads to some lysosomal inclusions, which are known as lipofuscin. Lipofuscin is a lysosomal inclusion, which is a hallmark of aging. And before we do this study, I must say that people knew, or many scientists knew, lipofuscin presence in the neuron, but not in microglia. In microglia, it was always neglected. But we could see that, especially upon aging and upon myelin removal by cells, this lipofuscin formation is going up and up. And also on top of that, cells become a bit slightly active. For example, they can produce, in aging, they can produce MHC class 2. And these characteristics are basically characteristic of a senescent cell, aged cell. Therefore, here we concluded that myelin overload leads to not only lysosomal inclusion, but also induce the age-related changes in microglia. This data, as I said, is a summary. This was published in Nature Neuroscience, and now we come to the second part of my title. which, as you can see, that in brain we have mainly, we have different regions, but in terms of myelin covering the brain, we have two regions. One is gray matter, which we have barely myelin there, and we have white matter, and which is actually quite full of myelin, yes. And what you can see here, and you can appreciate here, is that it seems that microglia in the white matter, when they are exposed to a lot of myelin, as you see here, they change. Basically, they become aged. They somehow, their functionality somehow goes down. Then with this, we wanted to understand what could be the difference of microglia responses in the white matter compared to the gray matter. Why cells are changing in the white matter, and if they are changing, what would be the comparison with the gray matter? Therefore, to understand this and to address this question, nowadays it's very common and quite easy to run single cell RNA sequencing, where you can basically isolate single cells, and then you can look for gene profile of each cell, and then you can cluster the cells, and then based on the difference of the gene profile, you can get different clusters, yeah? And this is what we did actually. We dissected the white matter, which was like optic tract or corpus callosum full of myelin, and also we dissected gray matter. And then we had two pieces of tissue. We send it for sorting with FACS, and we isolate microglia from both tissue. And then we did some different analysis with different techniques. One is SMART-seq2 and one is droplet-based RNA sequencing to, as I said, to basically analyze the single cell RNA or gene profile, basically. With that, we end up, we could basically analyze the transcriptomes of single cells through UMAP. I don't want to go through the details, but with that, with this analysis, we could distinguish the single cells from gray matter and also white matter. And with this data set that we got, we sort them, as I said, based on the gene profile, and we came up with 4 distinct populations in the brain, basically. As you can see here, 2 of them we called homeostatic number 1 and homeostatic number 2. And here is just a summary of our analysis, that these 2 populations are coming up not only in the gray matter, but also in the white matter. But interestingly, we had 2 very specific populations which are coming up only in the white matter. One of them we called activated microglia, and the other one we called white matter associated microglia. Next, it's important, of course, to see, okay, now we have clusters of the cell and we have 4 populations. How are they in terms of gene profile? Which genes are they really expressing? If you come with me and follow me, I can guide you through this detailed data that white matter associated microglia, they are expressing genes which are mainly relevant to phagocytosis or antigen presentation or processing, meaning that they are mainly involved in the activation of the cells, yeah? But then if you come to the end in the set 4, for example, you see that in white matter associated microglia, they don't express genes that are involved in during the homeostatic status of the cell. Like they don't have TMM119 or 6-TDCR1 or P2RY12 or 13, meaning that they are quite active, yeah? But then if you come to the activated microglia, again, you see that they're in the activation part, and also, again, they are not homeostatic, but mainly compared to the WAM, they are expressing genes which are related to the ribosome or protein translation, meaning that they might be important for the protein translation in the brain. And then completely vice versa, I must say there are two homeostatic 1 and 2 populations that they are not active. They don't express any activation marker. However, they're quite steady and they are expressing molecules which are relevant in the homeostasis. Yes, this was the gene profile of our 4 population. Okay, as I said, we were always interested in myelin and also in aging. Therefore, the next question was to know if these white matter associated microglia are really age dependent. For that, again, we dissect gray matter from the white matter and we isolate microglia and then send it for bulk RNA sequencing. This time was just the bulk sequencing, was not single cell sequencing. And there we came up with a huge amount of data, and there we analyzed the data and we compare the upregulated genes in the white matter old animal and compare it with the gray matter old animal and also with white matter young animal. Yeah, because we are interested only in aging. And then suddenly we came up with 39 genes that after this comparison, and these 39 genes are overlapped with our VAM signature because they are also expressing, they are also genes that are expressed, and we already showed in single cell RNA data that they are also expressed in white matter associated microglia. Some of them are like galectin-3, like CST7, which is important for the antigen presentation, and lipase, which is important for lipid metabolism. ITGAX and CREC7A also important for activation. Yes, that was already good news for us, but since we were also still, we wanted to prove that, we took advantage of the data set of other people, because as I said, nowadays, many people are doing single cell RNA sequencing analysis on microglia in different tissues, in different conditions, and also in different age. And for that, we took 2 publications, very important recent publication from Homand and Frigerio, where they basically did single cell sequencing, not only in age animal, but also in young animal. And then luckily in both data set, we could find again our 4 population, yeah? But interestingly here, we saw that the activated microglia and also the other homeostatic population are coming up no matter how old the mice are, yes? However, if you look at the VAM, it comes up only with time, especially here, for example, during the time, suddenly it appears. And with this, we could conclude that the formation of white matter associated microglia is basically aging dependent. Okay, that's quite solved. And now we wanted to dig a bit more in the gene profile of this VAM signature. What are they exactly? What they can be resemble, yeah? To what they can be resemble. What we did, again, we compare our data set with the data set of other people, especially with the data set that was published recently, in 2017, and it was a very important work from Karen Shaul, where they could show that especially in the AD or Alzheimer's disease model, a population of microglia, which is called disease-associated microglia, that nowadays we know it as DAM, coming up. But this activation, these cells are quite activated in AD model. And of course, this is now also in other models, has been in other neurodegenerative models has been also discovered, but those years just was AD. And then in a state, and this activation happens in 2 stages. One stage is TREM2 independent pathway, and the other one is happening through a TREM2 dependent pathway. TREM2 is a molecule which is important for the activation of, basically some people say it's a receptor for the anionic lipids, and it's also important for the lysosomal activity of the cell and also for, in general, for the activation. Anyway, I don't want to really go through that because TREM2 is also a molecule that now many people are working on it. But anyway, I just explain it here because this was important for us, that our, as you can see, our population VAMP, white matter associated microglia, and also activated microglia are expressing distinct parts of DAM2, the DAM which is in stage 2 and is TREM2 dependent. Yeah, as you can see, for example, this part is expressed also overlapped with here, but again, this part is overlapped with here. And yeah, distinct parts of the VAM is similar to DAM. Then next we wanted to know, okay, if we have similarity to DAM2, do this population also depends on the TREM2 or not? What we did to answer this question, we got aged TREM2 knockout mice, yeah, and then again we did the same procedure with the white matter and the gray matter dissection, and we did single cell RNA sequencing. And there we could see that, interestingly, in the white matter and in TREM2 knockout mice, the activated and VAMP are not coming up. And with that data, we could conclude that the VAMP is not only age dependent, but also depends on the TREM2 activation. Okay, next step, we wanted to know, okay, where are they in the brain, yeah? Where we can find them. For that, we took advantage of immunohistochemistry and also RNA-scope. And there, here I can help you go through. We had four markers, Galectin-3, CLEC7A, AXL, and 8GAX. These markers has been already published or established for recognizing DAM population, disease-associated microglia. And since we had similarity, we also took these antibodies. We stained the whole brain, and then we looked at not only cortex, which there is a bit of myelin, but also in the corpus callosum. And interestingly, we found that this population is mainly the population that express each of these markers, and we call them now VAM positive, are mainly expressed in the corpus callosum. As you can see in the, or let's say in the white matter, in this quantification, you see that we barely see any VAM positive cells. And in the gray matter, however, in the white matter, we have them, but we also see that they are kind of not really evenly distributed. Why? Because some of them are expressed with the through single microglia, and the microglia are single. However, some of them quite drastically are expressed in some clusters of microglia, which we call them microglial nodules. Then, as you can see, VAM are localized in the corpus callosum and also in the microglial nodules. Okay, what are microglial nodules? In aging brain of a mouse, and also, I have to say, in human, these nodules are formed mainly in the white matter and mainly in the corpus callosum. If you look at this box A in the cortex, you see that cells are quite singular. Yes, they are really, like, you see the nuclei separated. However, if you look at box B and C, which is in the corpus callosum, you see that often these cells get together. They make like a cluster, and then this structure known as microglial nodules. And we could quantify that the number of nodules is quite increasing in time and in aging. Interestingly, I can tell you that this population are also appear in the multiple sclerosis disease, for example. Okay, but what is the function of this VAM if they are in the corpus callosum, if they are in the microglial nodule, what they are doing? We correlate the microglial nodules in the light microscopy with the electron microscopy. What we could see is that in the electron microscopy, these cells are in the vicinity of some black structures which are already degenerated myelin or even myelin debris. Yeah, it seems that they are basically taking these fragments again. Therefore, we used our IBt-1 and MVP antibodies, and we looked at the corpus callosum, especially in the microglial nodules, these clusters. Yeah, and interestingly, we saw that they are indeed taking up some myelin fragments. And interestingly, again, we saw that half of the cells or microglia that are that have internalized MBP, they are in the nodules. Then next step, I just wanted to say something here that as you see, white matter associated microglia are formed in the corpus callosum. They are mainly in the microglial nodule, and they are again taking care of myelin fragments. Next, the question was that, okay, we say VAM is to form is depending on the TREM2. Then we wanted to know if we knock out TREM2, what will happen to VAM? Is it still forming or not? We took TREM2 knockout mice, and interestingly, we saw that indeed the TREM2 knockout mice cannot express any genes which are relevant to VAM. And also another interesting observation in these mice was that if you look at the TREM2 knockout mice, they are basically in the homeostatic state, meaning that they don't express, they express compared to the control age animal, they express a considerable amount of P2RY12 or TMM119. These are the main or known markers for homeostatic cells. Yes, meaning that in the normal control situation, cells are slightly active, but not in the TREM2 knockout mice. And again, we see that in the TREM2 knockout mice, not only we don't have any VAM forming, but also we don't find any microglial nodules in the corpus callosum. Cells are always in the singular, and they are very single, the individual way. But in the corpus callosum of a control animal, you can see a lot of these clusters happening. Then next we wanted to know, okay, in TREM2 knockout mice, there is no VAM form, there is no microglial nodules form, but we know that VAM is important for myelin removal. Therefore, what is happening to myelin in the TREM2 knockout mice? With this, again, we did some EM analysis, and we saw that indeed in the TREM2 knockout mice, myelin fragments cannot be removed. They are just floating around and in the extracellular space. And with time, the number of these extracellular fragments in TREM2 knockout mice is increasing. Indeed, then in TREM2 knockout mice, the formation of VAM and in turn as well, the removal of myelin debris is quite deficient. Next question was, okay, what happens now to microglia in TREM2 knockout mice when they cannot really respond to myelin fragmentation by forming VAM? Are they also changing? Yes, they are changing. Again, we did some correlative light and electron microscopy. As you see, these cells are not really normal, and also inside they have some black structures, which are, and we call them now, we call them inclusions, some lysosomal inclusions. And the number of these cells with the inclusions, we saw that is increasing with the in the TREM2 knockout mice. What could be that inclusion? We thought it can be myelin, because it seems that TREM2 knockout mice, they take up myelin, however, they cannot digest it. Then myelin get accumulated in the TREM2 knockout mice, and we have these inclusions. And also the cells, the morphology of the cells is changing. Microglia, as I said in the very, very beginning, they have this very characteristic structure of elongated branches, yeah, that they can go around and survey the environment. But interestingly, in our TREM2 knockout mice, we see that this regularity of the process is gone. and the process is somehow fragmented, all the processes of the cell. And we can, when quantify that, we see that the number of these cells, the microglial cells with irregular processes, is increasing in the TREM2 knockout mice. Okay, for now, we already learned a lot about VAM in the TREM2 knockout mice, and also in the steady state, that they are important for in the normal condition or normal aging. This VAM formation is important for removing the myelin fragments during aging. But how about pathology? It's always important now to know. Then we wanted to study the VAM population in the pathological condition. But which pathological condition? Of course, our first guess was Alzheimer. Why? Because we saw that the gene profile of VAM is similar to DAM, especially DAM stage 2, which was already known to be formed in the Alzheimer's disease condition. Yeah, this is why we wanted to know what would be the difference between VAM and DAM formation in AD, and what is the time sequence of this population. Then again, we did a single cell RNA sequencing, but this time in the 6-month-old FAD mice, which are a model of AD. And luckily we could distinguish our, we could detect our populations, homeostatic 1, 2, and also VAM, plus DAM, because anyhow now we are in the pathological condition, and of course we should detect also disease-associated microglia. And then we also compared our data set with the data set of people who have done with like Regio, that have done the who has done the RNA sequencing in another AD model, like APP model. And again, we could come up with our 4 population plus DAM1 and DAM2. But then we did some time analysis, and we realized that interestingly, already at 3 months of age, the VAM population appears. However, there is no DAM, and DAM comes next. And this was very interesting to know that in the time sequence, it seems that in the Alzheimer's disease, VAM comes first, and then it shifts to DAM. Then next would be, now that we know about the timing, now we want to know how they are different from each other in the Alzheimer's disease situation. With this, we again look at the microglial nodules, because as I said, VAM is in the microglial nodules, but this time we looked at the corpus callosum, where we have microglial nodules that are not associated with the amyloid plaques, because we know that in the AD model, amyloid plaques are formed. Okay, not mainly in the corpus callosum, of course, but in the cortex. But still, we were careful not to look for clusters of amyloid plaques. We looked only in the microglial nodules, and interestingly, we saw that these microglial nodules are again with a myelin inside. And we are sure that what we are looking at is really VAM population. And then this VAM population, not surprisingly, is quite active by expressing CLEC7A and also galectin-3. But more interestingly, as I said, already at 2 months of age, which is quite early for AD, especially in FAD mice, we have a lot of microglial nodules with the myelin inside. And this phenomenon is happening more even in 6 months. Yes. We also wanted to go ahead further and see what is really the difference between VAM and DAM. We looked at the gene that are differentially expressed in these 2 populations. And one of the best candidate that we had is macrophage migration inhibitory factor, because this, as you see, according to this data, here is the VAM. Yeah, if you compare it, and here is DAM, and it seems that in DAM we have we have less of MIF, but in VAM we have more. And this population was with this gene is important for us to distinguish these 2 populations. We compare this with another marker, which is CLEC7A, because it's basically everywhere in both populations. What we could see, we got a good antibody for MIF. And again, we did some staining, not only in the corpus callosum, but also in the cortex. In the corpus callosum, when we have clusters of microglial nodules that are not along with the amyloid plaques, we don't have any MIF. There is no MIF in the microglia in the nodule, yeah? However, they are expressing CLEC7A, they're active. But if you look at the amyloid plaques, they are MIF positive here. They are MIF positive, but also CLEC7 positive. Okay, this is also another way to show that VAM basically here is different than DAM. And since VAM and DAM both are known to be age depending, then we wanted to know if we can also recognize or detect MIF in the aging. And luckily, we couldn't find any MIF expression in aging. And this is also, again, shows that MIF expression is quite dedicated or quite specific to DAM, but not to VAM. Okay, with this all data set, we can say that VAM and DAM are coexisting in the brain of the AD model, but they are distinct populations. The last thing that we wanted to know is that how is the signaling pathway of formation of DAM and VAM in the aged model and also in the AD. We know that DAM formation is depending on the ApoE pathway. For that, again, we look at the data set of people which have done AD model crossed with the ApoE knockout mice. And there we looked at, again, our population. We detected that in this model, when there is no ApoE, there is a drastic reduction of not only DAM in black, but also VAM or population. But this was the data of other people. What about, we wanted to also prove this, what is happening in our data set. Therefore, we again took aged ApoE knockout mice, and there, interestingly, we saw that there is no change in VAM and activated formation, meaning that the formation of VAM is not depending on ApoE. This we could confirm in the immunohistochemistry when, again, we stain the tissue of the AD model, ApoE knockout mice compared to the control aged animal, and with the galectin-3, CLEC7A, and AXL. And we saw that in both ApoE and control, they are coming up. There is no difference, meaning, again, that basically this is really not depending on the ApoE. At the end, I want to just summarize our data that we could find a novel microglia subtype. We call them microglia, white matter associated microglia and activated microglia, that they are specific to the white matter. And we saw that the VAM and activated subtypes are both TREM2 dependent, but only VAM is age independent. And we saw that VAM formation is really not depending on the ApoE pathway. And interestingly, we could see that VAM gene signature is partially shared with the DAM gene profile, and they are coexisting. VAM and DAM, they coexist in the Alzheimer model, but they can be distinguished by the differential gene expression and also with their function. And our very important message here is that it seems that in aging, the activation of the microglia might be dominated by forming the VAM, and then it can be shifted toward DAM population when there is a pathology like AD, for example. Then we can say that it's a continuous phenomenon from start from VAM and goes toward DAM. And since we are coming from a myelin lab, that was also important to mention that it seems that the VAM formation is a protective response toward age-related myelin fragmentation. And with this, I want to thank you that you invited me for this talk, and I hope that you enjoyed it, and I'm happy to take questions.
Thank you very much, Shima, for your nice presentation. Sure. So if there are questions, either you can raise your hand and I will give you the opportunity to ask, or write it in the chat and I can read it. Carla, go ahead, please.
Hi,
Hi.
thank you for your talk. It was very interesting. And I was just, I don't know, a comment, like it is known that left-handed people has or have a larger corpus callosum compared with right-handers. So I think it would be interesting to see if there is any difference in the quantity or the function of this kind of microglia, the WAM, between left-handers and right-handers, especially also when aging or some dementia is present, right? Or what do you think about that?
Yeah, I don't know if I mean, you said that this is already known that between these two populations, that's interesting data, I would say. Yeah, that one can check, of course, but on top of that, it's also interesting to know that microglia are also different in different genders. The female microglia are slightly different than the male microglia. Therefore, I would say this can be what you are suggesting can be also something that yeah, one should take a look.
Okay, thank
But
you.
I don't have any more comment on that. I cannot really say. I mean, if there is, I know that, for example, with I mean, this is the ongoing research that with learning, you can improve myelin formation, for example. Yeah, while you are already adult and the myelin formed already completely formed, and while you are learning piano, no matter how old you are, you can trigger myelin formation in your brain. That's something that is really interesting. For me, it's fantastic research. It's ongoing.
Yeah, okay. Thank you so much.
Sure.
Any other question?
No sé, mi reina. Yo ahorita voy por el cilantro. El gas sí, pídelo ya de una vez, sí.
¿Pueden apagar sus micrófonos, por favor?
Por favor. Well, meanwhile, people. Oh yeah, Leonor, go ahead.
Shima, thank you very much for such a nice talk. I really enjoyed very much. As you know, with aging, the inflammatory signals are increased.
Sure.
And we know very much about all the signaling pathways controlled by the inflammation. Do you have any idea what is the role of inflammation in this new phenotype of microglia?
Yeah, I mean, in new phenotype of the population that I mentioned, or in general?
Yeah.
What is? We think that since they are coming only in aging, in the white matter, this population, it has something to do with myelin, because myelin is somehow broken. And we could also, in another publication that is still not published, we showed that myelin, when myelin is broken, they express, or basically they have, because myelin is quite membranous, yes, and it has a lot of in the inner membrane, it has a lot of phosphatidylserine, and when they is broken, it seems that this phosphatidylserine flips out, is like basically apoptotic cell body, yeah, and this somehow can be exposed to the cell, which are microglia. And we think that since we are coming from a myelin lab, we think quite strongly that this inflammation or slight inflammatory characteristics of microglia, especially in aging, has something very strong to do with myelin degradation.
Okay. Yeah.
Yeah. But of course, in general, it's already shown that microglia are activated slightly, or they call it primed, and slightly activated in aging, but nobody really took a look at the different regions to see where they are and how they are activated. Now, what we can say in our study is that they are activated mainly in the corpus callosum. Yeah.
Okay. Yeah.
Compared to the gray matter, what one also, for example, we didn't find this population in other region. In the striatum, we didn't find them, or in the hippocampus, we didn't really find them.
Yeah.
Then I think the yeah, they are important for this, and especially if you take a look at the data in the AD model, because you see that even in the AD model, in the very earliest stage of the disease, you have myelin inside these cells, meaning that the inflammation already starts there. But then when the disease comes, basically the inflammation gets bigger and bigger and then goes through the disease-associated microglia.
I see. Okay. Thank you. Thank you very much.
There is a question in the chat by Angel, and he mentioned that during aging and Alzheimer's disease, reactive oxygen species are elevated. So he asked whether you have know how this would affect WAM diversity.
We didn't really look at the ROS production in this population at all. Therefore, I really cannot comment on that. I mean, of course, how this can affect the WAM diversity, I would say, as I said, it's just a guess because we didn't really study that, but I would say these WAM population are not very active, because cells, especially phagocytic cells, they produce ROS when they are really aggressive. And I would say that maybe in the earliest stage, there shouldn't be any ROS. And yeah, maybe it comes later, but of course one should study, and we didn't do it.
There is another question in the chat by Eladio, and he asks, do you think the microglia is the only cell involved in white aging decrease, or thing like the astroglia is involved too, or other cells of the immune system?
That's a very, very good question. Yes, we know that astrocytes, sometimes they also can take over, and they can also, we already detected that in some other project, that astrocytes also can take up myelin debris when they are broken.
Enrique, adelante.
Gracias. Did you measure any of these phenomena in the spinal cord? Do you have any, do you think you can extrapolate this data from the brain to the spinal cord, or you think that there should be some difference? Because I think that metabolically is not going to be the same thing.
No, definitely. Yeah, that's a good question. Not that we really precisely did that. No, we didn't do it. But in TREM2 knockout mice, a colleague of mine has been doing or was doing some studies in spinal cord, and he also could repeat some of our data in the TREM2 knockout mice, but not in terms of WAM formation. This we really didn't do because there also the amount of myelin that exists is quite low, and it was quite difficult to get enough cells for RNA single cell RNA sequencing. That was kind of a technical problem, but yeah. But I would say nowadays with the advanced technology, one can also check in this way.
And what about multiple sclerosis, which is really a disease of the myelin?
Yeah, that's of course a very important question. I would love to continue the project on that area, because one of these, as I said, these microglial nodules that I also, along with the WAM formation, they have been, this data is not published, but some people, they detected even in the aging brain, human brain, yeah? And therefore, would be interesting to know what would be exactly again, when WAM really comes up, or when this disappears through the different stages of multiple sclerosis. But we didn't really check because, yeah, we were just working in the physiology so far.
Thank you very much.
Sure.
There is another question by Angel Flores, and he mentioned that in other publication, the endocannabinoid system reduced the brain injury and the associated white matter demyelinization in neonate rats. So in your opinion, how the endocannabinoid system have a function in aging?
I have to say, I have to see the publication. I just cannot comment on that, and I don't want to guess that how would, these endocannabinoids have a function in aging. Sorry, I don't know the answer.
Okay, thank you. Humberto Martinez mentions a bit of a nature-nurture question. Would particularities of the VAMP be related to an underlying signature of white matter population homeostatic microglia, or the particular stimulation profile that the white matter provides?
If I understood the question, okay. Can I also see it? Oh, yeah, okay.
Humberto, why don't you ask your question?
Particularities of VAMP be related to an underlying signature of white matter population homeostatic. I think they are not really related. VAMP not really related to homeostatic microglia, because basically they are losing their gene signature in the homeostasis. Or the particular stimulation profile that the white matter provides. Yes, of course, because as I said, white matter is quite affected in aging, and as for the previous question, I said that, for example, we know that the myelin, when myelin is broken, they also have some stimuli like phosphatidylserine that can kind of stimulate the formation of VAMP.
So I have a question, just waiting to see whether there is another one. In the TREM knockout mice, all TREM knockout mice, do they perform well in cognitive tests, better than knockout, than wild type, or are worse?
Actually, I think they are fine. These mice we got from a group in the same department, and I remember that one of the postdocs there, he was also doing some behavior tests, and he never saw something super drastic in these mice. I mean, especially in the aged mice, there were some slight changes, but they cannot really distinguish this is because of aging or because of TREM2. It's like they had a slight change and was not so. I remember we had this discussion and they mentioned this.
Thank you. So it seems that there is no other question. So first, thank you very much, Shima, again for your talk and for being here with us. And gracias a todos por asistir a pesar del cambio de horario. Realmente les agradecemos mucho su presencia. Thank you, Shima. And hopefully in the future you can really visit us.
Yeah, sure. I would be happy. Sí. Okay, gracias.
Okay.
Thanks.